Yes. So today's topic that we are going to discuss is transcription. So earlier we discussed DNA replication. It is a mechanism to synthesis DNA using the DNA as the template. So the two strands of the parental DNA is act as a template that is going to be replicated or create synthesis the complementary strand based on the DNA, that is called DNA replication. But what we are going to discuss today is transcription. What is transcription? Synthesis of RNA. RNA is going to be synthesized using DNA as a template. So here you can see the double-stranded DNA act as a template for the synthesis of RNA. DNA which is double-stranded, whereas RNA is a single-stranded. It is a molecular process in which RNA is synthesized using DNA as a template. So let me tell you some basic facts about RNA. So RNA is a polymer of ribonucleotides. What about DNA? DNA is a polymer of deoxyribonucleotides. But here we are discussing RNA. RNA is a polymer of ribonucleotides held together by 3' to 5' phosphodiester bridge, and they are single-stranded. DNA is a double-stranded, RNA is a single-stranded molecule. And it is the only molecule known to function both in the storage and transmission of genetic information and in catalysis. So the information lies in DNA are going to be, so DNA which is act as a template for the synthesis of RNA. The information lies in the DNA is going to be transferred to RNA, and RNA consists of codons which will code for different amino acids according to the nature of proteins. So all RNAs except the RNA genomes of certain viruses derived from information which is stored permanently in DNA. So mostly the RNAs are expressed based on the DNA template. So these are different kinds of RNA, that is mRNA, tRNA, rRNA. So it performs various functions. Say for example, mRNA is referred as messenger RNA that transferred information of gene to ribosome, that is encodes amino acid sequence. And tRNA, it reads codes on mRNA and transfers appropriate amino acid to mRNA. And rRNA is a constituent of ribosomes. So don't get confused. We will discuss about all these RNAs later. But today for our discussion, let us take the example of mRNA. mRNA is the messenger RNA. It takes the message of DNA to protein. So they are referred as messenger RNA. It transfers information of a gene to ribosomes, which will encode the amino acids. Okay, so to the focus of today's talk is all about only mRNA. So the synthesis of mRNA is called as transcription. So see, this is the diagram that shows the different types of RNA that's present in our body. Ribosomal RNA, that means the ribonucleotide present in ribosome. Then mRNA which is single-stranded, tRNA which is also single-stranded, but which forms some base pairing. That's why it has formed stem loop structure. Wherever it has got complementarity, it forms hydrogen bonding. Wherever it doesn't have the complementarity, it forms a loop-like structure. And altogether, it forms clover leaf models. It looks similar to clover leaf. That's why it's called as clover leaf model of tRNA. Then you have miRNA, that is microRNA. Then there are few RNAs called as ribozyme, U1 RNA, and so on. We will discuss each of these RNAs in later. But today's discussion part, we will take mRNA. So in case of DNA replication, the entire DNA molecule is going to be copied or getting replicated or used as a template for the synthesis of RNA. Say for example, you have a genome of 1 base pair to 3000 something like 11,000 base pairs, means all the 11,000 base pairs is going to be copied or synthesized in case of DNA replication. But what about mRNA or RNA synthesis? In case of transcription, a particular gene or group of genes are copied at any time and some portions of DNA are never transcribed. Say for example, you have RNA, you have a DNA molecule. In case of replication, the entire DNA molecule is going to be copied or replicated right from the first base pair to till the last base pairs. Do you understand? But what about transcription? What is transcription? Transcription is the process of synthesis of mRNA using DNA as a template. So in this case, not all the genes, or otherwise we say, not all the base pairs are going to be transcribed. See, DNA acts as a template. So RNA is going to be synthesized using a DNA as template. Now, where is the production or where is the synthesis of RNA? Now, you may get a question whether the mRNA is synthesized right from the first base pair to till the last base pair. The answer is no. The synthesis would not take place right from first base pair till the last base pair, whereas the DNA molecule is going to be transcribed at specific region. Such regions are called as gene, a segment of DNA that codes for a type of RNA. That is called as gene. Not the entire genome is going to be transcribed. Please understand, all of you. Not the entire genome is going to be transcribed. But what is going to be transcribed? You have different genes. See, look at the genome. This is the entire double-stranded DNA of an organism. Okay. This is the entire double-stranded DNA. So in this you have gene here and gene over here and another gene here. So the genome is the haploid set of chromosomes in an organism, or a complete set of chromosomes in an organism is called as genome. It includes the first base pair of the organism to the last base pair. But what about gene? Genes are part of genome. It may be small, it may be larger. Okay, but it's only a segment of DNA that codes for a protein is called as gene. So you would have studied the definition for gene in your genetics paper itself. Muton, recon, cistron, அப்படினு படிச்சிருப்பீங்க. Cistron the gene, a segment of DNA that codes for a protein. So in a double-stranded DNA like this, you have many genes. So according to the Human Genome Project, it was completed in the year 2003, and it has stated that human genome has 22,000 genes. Earlier it has reported 30,000 genes, but after so many research, now it has been proved that it has got 22,000 genes. Do you understand? So, genome is the entire DNA molecule of an organism which consists of many genes. What are these genes? Genes are the region of DNA that codes for a protein. See, our body has different types of proteins. Say for example, you have hexokinase, you have trypsin, you have pepsin, you have what? Hemoglobin, okay, and immunoglobin. There are number of proteins, enzymes are present in our body. Do you think all the enzymes are going to be similar? Definitely not. Not every protein is going to look alike. They are made up of the building blocks of protein, that is nothing but amino acids, but not in the same order, not gets folded into the same way. They are all having different amino acid sequences, and they fold in different forms. That is why it's from the single gene it has got forms different multiple proteins. Do you understand, girls? Gene abdina, gene that codes for a protein. If there are DNA segments that may not code for a protein, they are called as non-coding genes, a non-coding DNA. So coding DNA, non-coding DNA, there are two different things. When we discuss the prokaryotic and eukaryotic genome, you will understand. So there is a DNA called non-coding DNA. There is a DNA called as coding DNA. What is the difference between coding DNA and non-coding DNA? A coding DNA that codes for a protein, that's all. It codes for some protein. It may be insulin, it may be hemoglobin, or it may be any other metabolic, biochemical or molecular enzyme, even RNA polymerase or DNA polymerase and so on. Do you understand? And there are segments in the DNA which will not code for any type of protein, which will not involve in the transcription process. So they are called as what? Non-coding DNA. So all the coding DNA is called as genes. So in a genome, we'll have multiple genes in our body. See, from you would have studied about the difference between gene and alleles in genetics paper. Okay, so one form of allele is called as gene. So this from this region to this region, this is act as a template for the synthesis of one gene, one protein, and this gene that's going to act as a template for the synthesis of another protein. So you don't think that together as a double-stranded DNA is going to code. No, a single stranded DNA, because one template, one strand is enough to act as a template. Do you understand, girls? Are you all following?
Yes, ma'am.
Okay. Yes, ma'am. Right. So here is the differences between the replication and transcription. Maybe this we will discuss at the end of the class. Okay. So features of transcription. So it is highly selective. It is highly selective. What do you mean by selective? So in the case of replication, there is no selectivity that this part of DNA has to be copied first, or this part of DNA sequence should be copied immediately. No such selectiveness. Right from the first base pair till the last base pair, it's going to get replicated, and it starts at the origin of replication. Okay, but here transcription is the process of synthesis of RNA, and that too how it takes place. The RNA is synthesized using DNA as a template. So we have multiple genes in the genome. Each gene is going to be transcribed and forms RNA. Okay. So, and not all the genes are going to be transcribed at the same time also. Not all the genes going to be transcribed. Say for example, I can tell you an example of, what is it? Galactosidase. So, galactosidase, the enzyme that is involved in the hydrolysis of lactose. So when you grow E. coli cells in a medium containing glucose, then there won't be any synthesis of galactosidase. But when you change the medium, when you inoculate the E. coli cells in a lactose containing medium, immediately there is a synthesis of beta galactosidase enzyme. So only when there is a need. So again you will understand that the DNA synthesis process is a highly energy requiring process. So it won't waste much energy by synthesizing unwanted, unnecessary enzymes. Only when there is a need, it will be synthesized. So that's why it's called as it is highly selective process. Since this selectivity is due to signals embedded in the nucleotide sequence of DNA. Specific sequences mark the beginning and the end of the DNA segment which is to be transcribed. So you have some regions which will mark as a start region and end region of a particular gene which is to be transcribed. So this signals instruct enzymes where to start and where to stop transcription, and when to start, how often to start, everything is going to be determined by the signals.